Hey guys and welcome. Today we're going to be going over Edexcel AS Biology B revision. And we are doing Carbohydrates 1, Monosaccharides and Disaccharides. Okay. Biological molecules are the key to the structure and function of living things. Biological molecules are often organic compounds. Organic compounds all contain carbon atoms. They also contain atoms of hydrogen, oxygen, and less frequently nitrogen, sulfur, and phosphorus. Okay, so these are organic compounds. So remember in the previous video, we came across phosphate ions and nitrate ions, and we call them inorganic ions, and that's because they had no carbon atoms. They did contain the nitrogen and the oxygen atoms and the phosphorus and the oxygen atoms, but it doesn't count because it does not contain carbon as well. Okay. Most of the material in your body that is not water is made up of these organic molecules. An understanding of why organic molecules are special will help you to understand the chemistry of biological molecules, including carbohydrates, lipids, and proteins. Each carbon atom can make 4 bonds, and so it can join up with four other atoms. Carbon atoms bond particularly strongly to other carbon atoms to make long chains. The 4 bonds of a carbon atom usually form a tetrahedral shape, and this leads to the formation of branched chains or rings or any number of three-dimensional shapes. In some carbon compounds, small molecules, monomers, bond with many other similar units to make a very large molecule called a polymer. So once again, this bit should be GCSE, the monomers and the polymers and the carbon atoms making 4 bonds. The 3D shapes you won't have to learn for biology. This is just for A-level chemistry, but here they're just trying to give you a visual representation. The ability of carbon to combine and make macromolecules, large molecules, is the basis of all biological molecules and provides the great variety and complexity found in living things. Okay, so these shapes you won't have to learn. It's just trying to create a visual representation. Carbohydrates. Carbohydrates are important in cells as a usable energy source. They are also used for storing energy, and in plants, fungi, and bacteria, they form an important part of the cell wall. You will learn this later on. The best known carbohydrates are sugars and starch. Sucrose is the white crystalline sugar familiar to us all, while glucose is the energy supplier in sports and health drinks. You don't really need to know this. Starch is found in flour and potatoes, but the group of chemicals known as carbohydrates contains many more compounds, as you will discover. The basic structure of all carbohydrates is the same. They are made up of carbon, hydrogen, and oxygen. There are 3 main groups of carbohydrates with varying complexity of molecules: monosaccharides, disaccharides, and polysaccharides. Okay, so just heads up, mono is 1, di is 2, and poly means many. So that's 1 monosaccharide is 1 sugar, disaccharide is 2 sugars, and polysaccharides are many sugars. Right, monosaccharides, the simple sugars. Monosaccharides are simple sugars in which there is 1 oxygen atom and 2 hydrogen atoms for each carbon atom present in the molecule. So what they're talking about is, just to do a visual representation, so for 1 carbon atom, there is 1 oxygen atom and 2 hydrogen atoms. I've drawn it like this because later on we will see that it's over here. This is what they're talking about. Yeah, so that's a carbon, and it's connected, see? A general formula for this can be written CH2ON. Here N can be any number, but it is usually low. Triose sugars have 3 carbon atoms and the molecular formula C3H6O3. They are important in the mitochondria, where glucose is broken down into triose sugars during respiration. Okay, yeah, so you won't really come into contact with triose sugars here. but the important ones to focus on is the pentose sugars and hexose sugars. So pentose sugars, N5, have 5 carbon atoms and the molecular formula C5H10O5. Ribose and deoxyribose are important in the nucleic acids deoxyribonucleic acid, DNA, and ribonucleic acid, RNA, which make up the genetic material. Okay, so a pentose sugar would look like this. And you can see it says here ribose, and it has that shape. Hexose sugars and 6 have 6 carbon atoms and the molecular formula C6H12O6. They are best known monosaccharides, often taste sweet and include glucose, galactose, and fructose. Okay, so you would have come across glucose and GCC, and you would remember this formula as well. Now, 2 new sugars may come up to you, galactose and fructose. They were also monosaccharides. So they are also hexose sugars. So they would look like this. And so you can see that here. Molecular formula shows you how many atoms there are in the molecule and what type they are. But they do not tell you what the molecule looks like and why it behaves as it does. So what's telling you? So the molecular formula, if we're taking hexose sugars as example, O6. So they've told you it contains 6 carbons, 12 hydrogens, and 6 oxygens, but they don't tell you how it's arranged. So is it all a straight chain? Does it look like this? It doesn't tell you anything about the shape. To show this, you can use a displayed formula. Although these do not follow every wiggle and kink in the carbon chain, they give you a good idea of how the molecules are arranged in three dimensions. This can reveal all sorts of secrets about why biological systems behave as they do. Okay, so that's displayed formula. So it's when you display the shape. Alpha glucose and beta glucose. Glucose comes in different forms, isomers. So isomers may be a new word to you, and that's just when they've got the same molecular formula, but they may be arranged a bit differently. So including alpha glucose and beta glucose, these 2 isomers result from different arrangements of atoms on the side chains of the molecule. The different isomers form different bonds between neighbouring glucose molecules, and this affects the polymers that are made. Okay, so here, alpha glucose has the H atom on carbon 1 pointing upmost, whereas in beta glucose you've got the OH atom on carbon 1 that points upmost. That's the only difference. Here they've just drawn it, but they've removed the OH and the H's. In these diagrams, the positions of carbon atoms are represented by their numbers only. Note carefully the different arrangement of atoms around the carbon 1 atom in alpha and beta glucose. Right. Disaccharides, the double sugars. Disaccharides are made up of 2 monosaccharides joined together. For example, sucrose is formed by a molecule of alpha glucose joining with a molecule of fructose. And we know both here, fructose and glucose are monosaccharides, so 1 make 2. 2 monosaccharides join in a condensation reaction to form a disaccharide, and a molecule of water is removed. So condensation reaction, you will come across this in all biological molecules. That is how you join molecules together, through a condensation reaction. And condensation means so that when you come across the word, maybe you can see water, and that is why water is removed, and so you would see a water molecule as a byproduct. Yeah, so it results in a glycosidic bond, and this will be seen as this. So this is the 2 hexose sugars, and that is called a glycosidic bond. And it's a covalent bond because it is formed by non-metals. We use numbers to show which carbon molecules are involved in the bond. If carbon 1 on the monosaccharide joins to carbon 4, On the other monosaccharides, we call it a 1,4 glycosidic bond. If the bond is between carbon 1 and carbon 6, it is a 1,6 glycosidic bond. And what that's saying is basically, so that's carbon 1 over here, this is carbon 4. And so if these were to join together, that's called a 1,4 glycosidic bond. And they've shown that over here. And here it's alpha glucose produces maltose. And that's a 1,4 glycosidic bond. And you can see water is a byproduct. Okay, when different monosaccharides join together, different disaccharides result. Many disaccharides taste sweet. So sucrose is formed by alpha glucose and fructose, and is stored in plants such as sugarcane. Lactose, which is a milk glucose and it's found in milk, is by an alpha glucose and a beta galactose. Maltose is a malt sugar and is found in germinating seeds such as barley, and it is from alpha and alpha glucose. Right. This you would need to know. So Benedict solution is a chemical test for reducing sugars. Right, every sugar that you just learned is a reducing sugar except sucrose. So the Benedict solution is a bright blue solution because it contains copper ions. Some sugars react readily with the solution. When heated gently and reduced, the copper 2 ions to copper 1 ions will form a precipitate, giving a change from blue to orange. So when you heat a reducing sugar and it gives the color, it changes from blue to orange, you say that's a positive test showing reducing sugars. All the monosaccharides and some disaccharides are reducing sugars. Some sugars do not react with Benedict solution. They are known as non-reducing sugars. So sucrose would be a non-reducing sugar. So what you would do to confirm that it is sucrose that is present, you would then take the non-reducing sugar and add a few drops of HCl, hydrochloric acid, and that will hydrolyze the glycosidic bond. So actually what you would do, what would this do is once it reduces the bond, once it hydrolyzes the bond, you will have the production of alpha glucose and fructose. Those are monosaccharides and they are reducing sugars. So what would happen is the color would change. But after you've hydrolyzed the bond, you need it to cool down, and then you would add the sodium hydrogen carbonate, and that's because HCl is an acid. And you're going to put the hydrogen carbonate, which is a base, so a neutralization reaction. And so this produces the monosaccharide units of the sugar, we said the alpha glucose and fructose, which will now give a positive Benedict test, so that means it will change to the orange color. And that's it.